Seychelles warbler. So the credit for this work has to go to Dr. Ellie Chesterton, whose photo you can see here, who was a PhD student in my group, and she's now a software engineer. So cooperative breeding is when we have at least more than two individuals that are helping to look after an offspring. And the question is, why should an individual help? Because helping is obviously costly, and the individual has to forego their own reproduction to do this. And there's many hypotheses that have been developed about why individuals should help to gain direct fitness benefits. And the two that I'm going to focus on in this talk are the social prestige hypothesis, which was developed by Zahavi, which says that individuals are helping to advertise their individual quality and able to improve their future reproductive success. So, for example, enable advertising their quality to gain a mate. And the second hypothesis we'll look at is the breeding skills hypothesis, which says that individuals are learning breeding skills when they are helping, which then improves their breeding success in the future. So what's known about these hypotheses? Well, the most widely cited study is the Arabian babbler for the social prestige hypothesis. But there's a nice commentary by Wright, Jonathan Wright, which actually shows that in the first year when the social prestige hypothesis was suggested, it was probably actually more likely due to dominance interactions, and later work in that same population has found results which are not consistent with the social prestige hypothesis. Work in other species on the bell miner, the chestnut-crowned babbler, and the acorn woodpecker, looking at provisioning behaviour, has shown results which are not consistent with the social prestige hypothesis. And there's also been observations of false feeding behaviour in species such as the carrion crow and meerkats, which was also found not to be best supported by the social prestige hypothesis. So overall, there's very little support for this hypothesis in the literature. And the breeding skills hypothesis has most support from the Seychelles warbler. So this work was done by Jan Komda when he showed that female helpers, that when they went on to become breeders themselves, had short-term fitness benefits. In other studies in the long-tailed tits, they showed that helpers actually, after they'd helped, built lower nests than non-helpers, which had a short-term reproductive advantage in terms of the number of nestlings produced. But later work on this study showed that this didn't actually increase the number of offspring surviving to the first year. And in other species, such as the acorn woodpecker, the red-cocketed woodpecker, and the white-cheeked bee-eater, there have been no short-term benefits shown from the skills hypothesis. And in the lance-tailed manakin, looking at lifetime reproductive success, there was no breeding skills advantage for helpers compared to for individuals that had helped. And in fact, in the Western bluebird, helpers had produced fewer offspring the following year than non-helpers. So again, there's very little support for this hypothesis other than in the Seychelles warbler. So one thing that's important to point out here is that it's important to consider whether you're looking at short-term or long-term fitness metrics, because as I mentioned in the long-tailed tit, using the short-term metrics, there was some support, but not using the long-term lifetime reproductive success and survival to the first year metrics. And in lance-tailed manakins, looking at lifetime reproductive success, there was no support. So what we did was we tested these two hypotheses in the Seychelles warbler, and we have a fantastic data set on this island of Kuzan that you can see here. And it's really great for looking at fitness benefits of behaviours like helping, because it's a closed population. So it's quite rare for a wild population that we can accurately assign survival and lifetime reproductive success, because birds don't disperse from the island, and we monitor the entire population. So this is an aerial shot of Kuzan Island, and as we monitor the entire population, there's about 115 territories, and the whole island is saturated, so all areas are suitable for the warblers are inhabited. And in each group, there will be a dominant male and female, and subordinates of both sexes. And these the subordinates can actually be helpers or non-helpers, and both males and females can help. So we can identify these birds using the colour rings that we give them on the inside of their legs. And we assign helping behaviour through 1-hour nest watches when we see which individuals provision the offspring. And we use genetic data to be able to assign parentage to know who the helpers are and who the dominant breeders are. So as I said, in the Seychelles warbler, it's previously been shown that female helpers had faster in their first breeding attempt as a dominant. They built stronger nests and they incubated more, which means they were less prone to predation. However, this was before we had DNA data, and we've shown after this that 44% of female helpers could actually co-breed. So rather than helpers gaining breeding skills through helping, they may have actually gained direct skills from breeding. So what we did was to look at non-helpers and helpers, and we predicted that helpers would have an increased likelihood of becoming a dominant breeder, which is a test of the social prestige hypothesis. We then looked at whether or not we predicted that helpers would become dominants at an older age and have a shorter dominance tenure because they are spending time helping. Testing the breeding skills hypothesis, we looked at whether or not individuals that had helped produce more adult offspring, so surviving to the first year as dominants than non-helpers. And we also investigated more long-term metrics of lifetime reproductive success. And the prediction here is that helpers should have slightly lower lifetime reproductive success because helping and non-helping are evolutionarily stable strategies. And helpers gain some small indirect fitness benefits from helping. So what did we find? We found no support we found there was no statistical significant difference between helpers and non-helpers in the likelihood of becoming a dominant breeder. So that does not support the social prestige hypothesis. We did find that helpers acquired dominance at an older age because they spend time helping. And so you can see here, we've got helping on the x-axis, whether they helped or not before dominance, and age at dominance on the y-axis, looking at both males and females. And helpers take just over a year or so longer to gain a dominant position than non-helpers. And this analysis is actually controlling for co-breeding. So only females co-breed, and those females that co-breed also took longer to attain first dominance. But interestingly, we found there was no significant difference in the tenure of dominance for helpers and non-helpers. And I'll come back to this in a discussion. So testing the breeding skills hypothesis, we found that helpers did not produce more offspring during their dominance tenure than non-helpers, which is not supportive of, was not consistent with the breeding skills hypothesis. But what we did find is that individuals that co-bred before dominance produce more recruits as a dominant. So those females that helped before, that co-bred before, produced more offspring. So this suggests that Jan Conde's previous results were in fact due to co-breeding effects. So we found our results were not consistent with the breeding skills hypothesis. And then looking at lifetime reproductive success, we found no statistically significant difference between helpers and non-helpers. But we did find that individuals that were females that had co-bred before had higher lifetime reproductive success than those that did not co-breed. So what does this mean in terms of support for the literature? Well, we found our results were not consistent with the social prestige hypothesis. And this joins what is widely found in the literature now from the other species of limited support for the social prestige hypothesis. And then looking at the age at which individuals attain dominance and the tenure of them, we found that helpers attain dominance at an older age and have similar dominance tenures than non-helpers, which suggests that they live longer. So we did a sub-analysis of this, and we found, in fact, that the dominant females that were helpers did have a longer lifespan than dominants that had never helped. And this is actually consistent with previous work in the Seychelles warblers, which showed that individuals that have a delayed age of reproduction have delayed survival senescence, which supports the disposable soma hypothesis. Intriguingly, our results were not consistent with previous work on the Seychelles warbler. So we found no support for the breeding skills hypothesis. And this was due to accounting for co-breeding females. So the results previously we suggest are due to breeding females that have, helpers that have co-bred, gaining direct breeding skills themselves. And this then joins the literature where overall there's been no support for the breeding skills hypothesis. So in conclusion, we found that helping was not associated with long-term reproductive success. Our results were not consistent with the social prestige hypothesis or the breeding skills hypothesis. We showed that it's very important to separate out co-breeding from helpers, which is why our results differed from earlier work on the same study system. And it also highlights that it's very important to look at both short-term and long-term fitness proxies to understand where any benefits from helping behaviour may be playing out. If you're interested in this work, that you can scan here for the preprint that's available on the Evo preprint server. So thanks for listening, and I'm happy to answer any questions that you have.
Thank you, Anna. That was really cool. Any questions, anyone? You can hit the little raise hand button at the bottom if you have a question.
Hi, great talk. I was wondering, when you say co-breed, do you mean that in the same nest, the eggs are like laid by the dominant female and the helper both? Yeah,
that's generally the case, yeah. So they usually just lay one egg. Sometimes they'll lay up to 3 eggs, but those nests could have an egg from the dominant female and from a female helper, which is then co-breeding.
Okay, I had another question. Since you have genetic data, I was wondering what is the angle of kinship in this system? Like, are they more likely to help their kin?
Yeah, so the rule of thumb is that females and males will tend to help the female who was present that was feeding them as an offspring. So it tends to be their mother that they're helping. There's a small number of individuals that will disperse to another territory and help in a non-natal territory, but that's it's about 90% of helpers, and they tend to be females, and they then can gain co-breeding experience and tend to go on and inherit that territory that they're helping in.
Great. Thank you.
Thanks.
Jeff has a question.
Oh, Jeff, yeah.
I hope you can hear me.
Yeah.
All right. Thanks for the lovely talk. Can I just ask, I know you seem to be focused on direct fitness benefits. Did you investigate any other direct fitness benefits like group augmentation effects, or maybe that's down the road for you and your group?
Yeah, we haven't explicitly tested the group augmentation hypothesis. So Jan Commodore has looked at it in earlier work, and it suggests that if you're in a group with one helper, that has positive effects on the number of offspring produced. But if you have more than two or more helpers, it has a negative effect on fitness. So overall, we think there's limited support for the group augmentation hypothesis, but it'd be nice to revisit it with the larger data set that we have now. That's a good question.
Cool. Thank you, Anna. That was great. Okay. I think we're right on schedule. So our next speaker is Nicole Lopez. If you want to share your slides.
Okay, can you hear me?
Yep.
Okay, great. Just gonna. Okay, great. Yeah, so my name is Nicole Lopez. I am a PhD student at the University of Montana, but today I'm going to be talking about work I did during my master's at California State University, Long Beach, looking at evidence for a relative reduction in brain size when bearing large sexual weapons. Today's presentation, we're going to introduce sexual weapons and look at some studies to see whether or not they are costly. Walk through my methods of how we were able to measure relative investment in sexual weapon size, and then go after a question to see if there is an evolutionary reduction with relative brain size when bearing large weapons. First, to introduce our study system, animal weaponry. Animal weapons are morphological adaptations that arose either due to natural selection as a result of strong predation risk, and that form of weaponry is called morphological defenses or defensive weaponry. They are found across multiple different phyla and take on a bunch of different forms, and they're usually associated with the predator that they are pressured from. So a quick example is the spines of a spiny tenrec. Whereas the other form of animal weaponry is considered sexual weaponry, is selected by intersexual competition or competition between the same members of the same sex within a species to either possess, gain, or maintain mating opportunities. And this is going to be the form of weaponry we will be focusing on for the remainder of the talk. These sexual weapons are also found across multiple different phyla, taking on a bunch of different morphologies, having a bunch of different functions. Rather that be pure combat where the males entangle each other to gain resource to the females or mating opportunities, or that they serve more as a sexual signal and their status and size of their weapon is enough to dissuade fighting. These forms are also incredibly male biased. There are evidence of females bearing sexual weapons, but most of the time they are reduced forms and the males are under more directional sexual selection of having these really elaborate and intense sexual weaponry. Weapons in both forms can be very costly, not always. There is evidence that sometimes they are able to compensate through neighboring structures and not endure as much cost as we assume or expected. However, there is a lot still evidence that the growth of and the burden of bearing, developing, and yielding these morphological adaptations can be very energetically costly, and they can lead to a bunch of different types of physiological or behavioral trade-offs. So I'm highlighting a few studies here. So they found males that bared larger weapons performed less and produced less smaller nuptial gift sizes to the females during mating. There's also been evidence of reproductive trade-offs, so reduced testes size with males that bear larger weapons. And then focusing on here with relative brain size, they found that mammals that invested more in weaponry, they had relative reduced investment in larger brains. Taking that a little bit more in detail, this was a study done looking strictly at defensive weapon-bearing mammals. So these are animals that have these traits that were selected for increased survival under high predation risk, and they were able to quantify a defensive score. So that's their ability to measure how much investment these mammals were putting into their defensive weaponry. And then they used the measure of encephalization quotient, or EQ, as a measure of relative investment in brain size. And they found across all the species that they measured, there was a significant negative association between the two investments, suggesting some type of trade-off. And so this study, along with what we do know about sexual weapons, has inspired my question whether or not this similar trend of a reduction in brain size is also present when you invest more in sexual weaponry. And so hopefully we're going to show and predict that as males invest more in larger weapons, they will produce or result with a reduction in relative brain size. My study system, sticking with the mammals, specifically looking at ruminants, I'm going to be talking about 3 different groups. So we have first our tusks. So if you haven't heard of tusked deer before, they are found across 3 different families, Trabulidae, Moschidae, and cervids. These are enlarged upper canines. These weapons are strictly used in sexual combat, so they have low signaling power. They're mostly used for physical entanglement, where eventually the male who loses will run off, and that is how dominance is established in these types of groups. This is also the ancestral form of weaponry in deer, whereas most of the time when we think of deer weapon, we think of antlers, which is the derived form of weaponry in deer. These forms have multiple different functions compared to their tusked relatives. They have the potential to be used in intersexual threat displays where they can dissuade the other male from either engaging in fighting. And a lot of times fights are rare in antler-bearing species. However, they still are an honest signal of fighting. And lastly, the family Bovidae, we're looking at horns, which is the sole form of weaponry in this family. For the males, they are directionally selected for sexual weaponry. And then there's some kind of presence absence of females bearing the weapons as well. However, they are reduced forms and used in different kind of functions as compared to the males as a weapon. So these are the three groups that we are focusing on for this project, leading into our hypotheses and predictions. So our big hypothesis is that increased investment in larger sexual weapons leads to a compensatory reduction in investment in relative brain size. We have two different predictions. One, we're looking to see if males have a direct trade-off, whether or not males that grow large sexual weapons will have relatively smaller brain. So looking within the sex of males alone, since they are, again, male biased, they likely suffer greater reduction than compared to their conspecific females. However, what makes our study a little bit different from the defensive weaponry study is that our species are sexually dimorphic. The females either have reduced or no form of the weaponry at all, which allowed us to test another prediction of whether or not relative investment in brains is sexually dimorphic when males are investing in large sexual weapons. So we're hoping to find that as males invest in weapons compared to their females, there will be a relative reduction compared to their females in brain size as well. How did we perform this project? We were able to utilize 6 natural history museums where we were able to measure 413 specimens over 29 species. We measured both adult males and females, along with these morphometric that we have here represented of skull length, skull height, and skull width. We were able to also measure weapon length. So here's the first example of canine length. We take it from 2 measurements because these traits are very curved, and we wanted to create a better representation of the size of the weapon. So 2 measurements averaged, and then the maximum length was included. While we were there, we were also able to measure intracranial volume, which we were later able to convert into brain mass to later create a relative brain size quotient. Following with the weapons, we have horn length, also same method, 2 measurements averaged and the maximum value was used. However, the antler length, we were able to just take the longest length of the antler itself, represented here in red. Now that we have all of our raw measurements for all of our specimens, we are able to now calculate and predict relative brain size for each individual before moving on. So following some methodology from Body et al. 2012, which has been widely used as a way to measure relative brain investment or encephalization quotient, which I will be now calling EQ. This is when you take the relationship between brain mass and some type of body measurement, which we are using here as skull length because it was individually specific rather than species averaged. You take the relationship between those two and you're able to take the outputs and calculate a predicted value of what that trait should be based off its body size. And then you could take the ratio of that actual value and the predicted value, and you have a relative size. So if values are falling along that 1-to-1 ratio of EQ of 1, represents that they have a proportional brain size for their body size as predicted by that regression. If it's above, EQ is higher than 1, it represents that they have a larger brain than expected for their body mass or body size. And then also below, if they have a smaller EQ than 1, then they have a smaller brain than expected for their body size. So this is how we were able to calculate relative brain investment, and then following the same exact methodology, just changing up the y-axis. Instead of having brain mass, we used weapon length, and now we're able to also represent a relative investment in weapon size. So now that we're able to convert all of our values into some type of form of investment, we can now average the males and females for each species and run the analyses that we are interested in. So first, we are going to measure or run an analysis of female encephalization quotient or brain size investment with weapon investment, as we were able to collect canine length and horn length of the females of those tusked and horn species, and of course the female caribou or reindeer, what you know it as. And weapon type was included as a factor in our models. We followed the same analysis for males. So what is the relationship between male investment in weapon length and brain size, sorry. And then testing our sexually dimorphic analysis, we were able to do the the difference between male and female investment in EQ and the difference between male and female investment in WQ. So if it's a more positive value, that represents that males are larger. If it's more negative value, represents that females are larger. And of course, we ran this through a phylogenetic generalized least squares test to correct for phylogenetic relatedness, resulting in the tree we have here on the right. This is a representation of a few of our species that we were able to study in this project. Yellow is going to represent tusks, pink is going to represent horns, blue is going to represent antlers solely, and the black is just to represent those dual weapon species that bear both antlers and tusks. All right. So a reminder of our prediction. So prediction 1 was that species where males invest more in large sexual weapons, they will have relatively smaller brains. So this is hopefully what we're going to see. However, when we ran our analysis, both when looking at the males, which was mostly what we were interested in, we also looked at the females as well. We were not able to find any relationship between relative brain size and relative weapon size, suggesting no relationship between the two traits. So unfortunately, we were not able to support that prediction. However, it's led to a lot of more exciting ideas that we have going forward, which I will shine back in at the end. Prediction 2 was looking at that sexually dimorphic relationship. So do males have smaller brains compared to females in the same species when they invest more in larger weapons? We're hoping that as males get larger weapons compared to their conspecific females, they will have smaller brains compared to those females as well. And that is exactly what we see. So for our result here on the y-axis, we have brain sexual dimorphism, where the more positive the value is, males would have larger brains, more negative, females would have larger brains. If it's at 0, represented by that dashed line, that would mean they have about equal size investment in brain size. And as you can see, most of them fall under the 0, suggesting females have larger brains. And also as males get larger weapons than females, we have a negative relationship. We were able to support that relationship. However, there is still more to learn within these groups. As you can see, the antlers have higher EQs than the horns and the tusks, which allows for also more exploration into what these could mean. So that is the conclusion of our project. Our main finding was that males suffered a reduction in relative brain size compared to their conspecific females as they invested more in larger weapons. Since we did not find that directional trade-off or reduction in brain size, it has led to a lot of different ideas of whether or not these males are able to get around a trade-off by having more investment in structural support of neighboring traits such as skull length, shoulder height, or shoulder width, musculature. Maybe there's a potential conservation for relative brain size when you have a higher signaling power in the antlers and the horns, or there's trade-offs in other different pathways that are expensive, such as fecundity, or any other real pathway that's expensive. That'll lead to more questions to be answered. But that is all that I have today. I'd like to thank the Mammal Lab, especially Victoria Luce, for her help with data collection, my funding, RISE MS PhD program, help with statistics, and my committee members and all the museums that allowed us access to their collections. And with that, I will take any questions.
Thanks, Nicole. That was really cool. Any questions from the audience? You can ask in the chat or raise your hand if you have it. We have 1 more minute, maybe. So that sexual dimorphism result, I think, is really, really cool. Do you know, like, when you look at the relationship there and you have like a strange instance, like, for example, someone with a 1 score on their weapon sexual dimorphism, or a species, I should say, that has a much bigger brain size in males or females? I'm wondering if ones that really don't fit the line might like inform your intuition a little bit. Like if you look at them, there's something strange about them or something like that.
Yeah, absolutely. Especially this one that's high up here with the males being larger than the females, but still having about a 1-to-2 ratio of their sexual dimorphism. I believe that's moose, which I've looked into, that moose have smaller weapons than they should for their body mass. So there might be more to explore within that species alone of what they could be benefiting from conserving like a larger brain or not having as large antlers as they should. And that could be kind of based off habitat, or because moose have really, really large, heavy weapons, there's maybe just like a certain type of physiological limit as well there. But it's really exciting to kind of look through the antlers because they are deciduous as well, which could kind of give us some more information about their energetic trade-offs between horns and tusks, because they're permanent versus they have to shed them every year. So there's a lot definitely to explore.
Well, it's cool because the departures might help inform your intuition or your hypothesis building, right? Yeah, it's pretty neat. Okay, thank you.
Yeah, of course. Thank you.
So our next speaker is Kirsten Ferner. Do you want to share your slides? Sweet.
All right. Okay, my name is Kirsten Ferner, and I'm working as scientific employee at the Museum für Naturkunde in Berlin. And today I will talk about the development of the bronchial tree and the gray short-tailed opossum. In recent mammals, we have three infraclasses: the monotremata, the marsupialia, and placentaria. These three mammalian groups differ mainly in their reproductive strategies. In monotremes, the monotremes, which split 220 million years ago from the mammalian lineage, lay eggs, incubate them, and after an incubation period, a highly immature hatchling emerges from the egg. This reproductive strategy might resemble that of the common mammalian ancestor. Within the Theria, placentation and with that viviparity evolved, and the Metatheria and Eutheria separated around 140 million years ago. In recent marsupials, vitelline placenta evolved, and which allows only for a short gestation period and the birth of a highly immature, nearly embryonic neonate. During the early postnatal period, the young are firmly attached to the permanently attached to the teat, and most of their growth and development occurs postnatally, supported by a prolonged lactation cycle. In placental mammals, Coio allantis placenta evolved, which allows for a longer intrauterine development of the fetus. The neonates are always more developed compared to marsupial neonates. We find a wide range of altricial species to precocial species with intermediate stages existing. Typical altricial species like these newborn tree shrews are born naked with closed eyes, stay immobile in the nest, and are highly dependent on maternal care. On the other side of the spectrum, we find precocial neonates like the hare, which are born with open eyes, fully formed, and able to run. If we take a look on the life cycle of mammals, it becomes clear that the time immediately after birth is the most critical stage in the mammalian life. In the prenatal period, the fetus is protected by the maternal uterus and gets all what it needs from the maternal placenta. After birth, the nourishment is also provided by the mother by milk, and after sexual maturity, the animals are able to care for themselves, to feed and protect themselves. At birth, it decides whether the organism is capable of surviving. That means if all organ systems necessary for survival are functioning. An essentially important organ in this context is a lung. The survivability of the neonate depends highly on the maturity of the respiratory apparatus. The lung maturity in neonates seems to be variable. However, the process of mammalian lung development follows a clear pattern, which I will point out next. The structural development of the mammalian lung is well studied mainly in eutherian species like mouse, rat, and humans. After the embryonic phase, the pseudoglandular period follows, then the second canalicular period, and later on the sacular period, and finally the alveolar period. The three early stages of lung development fall always in the fetal period in eutherian mammals. Only altricial species, like the golden hamster, for example, are born with lungs at the late sacular period. But they reach the alveolar stage short after, in 2 days in the case of the golden hamster. Most placental mammals, including all precocial species like this guinea pig, are born at the alveolar period of lung development. In contrast, marsupials are born at earlier stages in lung development, the canalicular period, as it is the case in the gray short-tailed opossum, or in the early sacular period, for example, in the Damabala bee. Some kangaroos are a little bit more developed. In any case, the lung needs to be adequately developed in utero to function as gas exchanging organ. To fulfill the task of gas exchange, the lung has to have a very large surface area possessing thin air-blood barrier, a major surfactant system facilitating inflation stability, and an effective vascular system leading the blood to the gas exchange area, and finally a tree-like system of conducting airways ventilating the gas exchange area, the bronchial tree. And this study focuses on the development of the bronchial tree in the gray short-tailed. We used the gray short-tailed opossum, that's the Dividifer marsupial from Brazil, to investigate the development of the bronchial tree. The gray short-tailed opossum, Monodelphis domestica, resembles both the mammalian and the marsupial ancestor, and is therefore suitable to inform on the evolution of the mammalian lung. It offers a unique opportunity to studying the mammalian bronchial tree because, in contrast to Eutherian mammals, the formation of the bronchial tree occurs postnatally in a ventilated functioning state. Since the adult lung structure of the gray short-tailed opossum and that of Eutherian species resemble each other, it can be hypothesized that the development of the bronchial tree follows similar developmental trajectories as described for the Eutherians, however decelerated and postponed in the postnatal period. Current study uses X-ray computed tomography to three-dimensionally reconstruct the bronchial tree of Monodelphis in order to reveal the timeline of morphogenesis during the postnatal period. For this purpose, gray short-tailed opossums were obtained from a laboratory colony established at the Museum für Naturkunde in Berlin. In total, 33 animals from neonate ranging to 57 postnatal days and adults were included in the study. Coming to the methods that we applied in the study. Young ranging from neonate to day 28 were decapitated to allow better lung fixation via the trachea. And the whole animals were immersed in Karnofsky fixative. Older stages, from 35 days to adults, the lung was fixed with Karnofsky by means of installation fixation via the trachea, and the lungs were finally dissected. For visualization of the lungs in X-ray computed tomography, the specimens had to be stained in advance with tungstic acid. The whole specimen or the lungs were scanned with microtomographic techniques, and the 3D volume processing was done with the software volume graphics. The micro-CT data were analyzed as serial two-dimensional sections, and the bronchial tree was reconstructed. So it was marked, you can see it here in blue. And finally, a 3D reconstruction was done. So finally, the 3D reconstruction was transformed into a centerline model that visually shows the generation of the branches and allows to calculate mean, maximum, and number of branch generations. Coming to the results. The reconstruction of the lung anatomy in Monodelphis reveals that the gray short-tailed opossum has 6 pulmonary lobes. 4 lobes in the right lung, that's the cranial lobe, the middle lobe, the accessory lobe, and the caudal lobe. In the left lung, we have 2 lobes, the middle lobe and the caudal lobe. From outside, the left lung appears unlobated because the middle lobe and the caudal lobe are not separated by a fissure. But the bronchial tree indicates that there are 2 lobes. A literature survey of the bronchial trees in mammalian species reveals that in 21 species, among them, so these are marked in red here, among them most of the marsupials, one monotreme, the echidna, and in many placental mammals, the same lobation pattern is present as in Monodelphis domestica. The most common and probably plesiomorphic bronchial pattern in mammals is that what we find in Monodelphis domestica. These drawings show the major bronchial tree of Monodelphis from the ventral and dorsal view. The bronchial tree consists of a system of lateral row bronchioles, L1 to L4, a ventral low bronchiole, which is supplying the accessory lobe, and they have medial row bronchioles, M1 to M3, dorsal row bronchioles, D1 to D3, and finally terminal bronchioles, which forming the tip of the bronchial tree. The branching pattern of the bronchial tree is similar to that described in the mouse and appears to be stereotyped. 3 local modes of branching are used in 3 different sequences in the developing lung. These branching modes are domain branching, planar bifurcation, and orthogonal bifurcation. Domain branching means that the secondary branches are created by a row at different positions around the circumference of the primary bronchus. They are numbered in the proximal distal order in which they form. So for example, for the lateral branches, means that the first one to be formed is L1, the middle lobe bronchus of the left lung. Then the next one is L2, L3, and finally L4. Now I will show you the 3D reconstruction of the bronchial tree. Bronchial tree in the neonatal lung is characterized by short lobar bronchioles, which branching off the main bronchus, which open directly into large terminal air spaces. So this is a trachea, which branches to the, here's a bifurcation, the main bronchus, and that's the first lobar bronchus, which goes off and opens directly into the large terminal air spaces. By 4 dPn, the lung has entered the sacular stage of lung development with numerous large terminal sacs. First, short segmental bronchioles can be seen. The newly formed segmental bronchioles are smooth-walled and lined with respiratory epithelium. It's a little bit difficult to distinguish really between the air spaces and the new formed segmental bronchioles because all have respiratory epithelium. With 7 dPn, a more differentiated bronchial tree is present. In all lung lobes, segmental bronchioles are formed, giving rise to several subsegmental bronchioles. From 11 to 21 dPn, the lung is still at the sacular stage of lung development, however, increases markedly in complexity. This is accompanied with an increase in sacular number and decrease in sacular size. By 14 dPn, a functional bronchial tree has been established. Segmental bronchioles, often first, second, or more generations of subsegmental bronchioles, are present in all pulmonary lobes. Starting with 28 days, substantial changes take place in the lung Monodelphis domestica. The lung is at the transition from the sacular to the alveolar stage. Beside saccules, which are still present here, first alveoli can be identified at 28 days, and even first respiratory bronchioles can be detected. At 35 dPn, the lung has fully reached the alveolar stage of lung development. No saccules are present anymore. The structural development of the bronchial tree seems to be completed. All segmental bronchioles and subsegmental bronchioles in all lung lobes are present. And finally, also the structures of the terminal airways, like respiratory bronchioles and alveolar ducts leading to alveolar sacs, can be seen. By 49 and 57 days and in adults, no major structural changes can be observed. The major bronchial tree is still extending by forming new bronchioles, meeting the requirements of the growing lung. At 57 dPn and in adults, most easy to see, but also starting with 35 dPn, a special phenomenon can be seen in marsupials, which is unique to marsupials. The alveoli can be seen located in the walls of large terminal airways, like these segmental bronchioles, for example. The alveoli located in the walls of large airways might be remnants of the transformation of former respiratory epithelium into bronchiolar epithelium, and may result from the postnatal formation of the bronchial tree in a functioning lung under ventilated condition. And yes, these centerline reconstructions showing generations of end-branching bronchioles from neonate to 57 dPn. The colored circles indicate generations of end-branching airways, and which are explained here in the color bar. In neonates, the number of branches varied from 1 in where the lobar bronchioles open directly into air spaces, to 4 in the caudal lobes, where short segmental bronchioles went off. During the postnatal development, the median and maximum generation and number of branches increased steadily, visually shown by change of color from blue to green, yellow, orange, red, and finally purple. And at the time alveolarization begins, at 28 dPn, the number of branches increased markedly, almost doubling. And the final bronchial tree is present at 49 to 57 dPn. Up to 18 generations of bronchioles extend to the lung periphery of the caudal lobes. Coming to an end, I will now conclude the results of the study. The bronchial pattern of Monodelphis domestica, 4 lobes in the right and 2 lobes in the left lung, is common in other basal taxa and many distantly related mammalian groups. Therefore, the bronchial tree of Monodiathus could be considered as plesiomorphic to Mammalia. In marsupials, the development of the bronchial tree from an embryonic developmental degree to final adult lung structure takes place in the postnatal period in a functional state. Similarities to the branching morphogenesis described in mouse and humans suggest that this process is highly conserved, but follows similar patterns as described in placentals. Lung maturation in general and the branching morphogenesis in particular are highly conservative within mammalian evolution and are obviously controlled by a genetically encoded superroutine. I thank you for your attention, and yes, I'm looking forward to your question.
Thank you, Kirsten. I think in the interest of time, we'll have to forego the questions and move on to the next talk. But if you want to write a question in the chat or send a direct message to Kirsten with any questions, that would be great. Thank you. That was a really cool time series. Amazing. Okay, our next speaker is Peter Hood. I hit the ask to unmute on you, but I think you'll have to click something.
I thought I had.
There we go.
Yeah,
There we
okay. Right. So I lost a little time there, but anyway, I want to talk today about a family of.
You want to share your slides real quick?
Yeah, I'm working on it. It says that it is disabled.
You should have the rights to do it. You are okay. Try now.
There you go.
There we go. Sorry about that.
That's all right. Yikes. Oh, what am I doing? This is a mess. Seriously losing time here. Okay, thanks for joining me. I hope everybody can hear me now. I'm going to talk about the implications of presumed South American endemic in the early Paleogene North America. It happens to be a flightless bird. The family Forest Racidae, more popularly known as terror birds, are members of the order Caryamiformes, and these were apex predators throughout most of the Cenozoic in South America. They're survived today by 2 species of Seriamas, and these are somewhat smaller than any of the Forest Racids, but they are indeed predators. And slide's not advancing. There we go. We can see here on the lower left that the Forest Racids vary quite a bit in size relative to that human there. And the Seriama, the modern Seriama, the genus Caryama, is highlighted in green there. The smallest of the Forest Racids is Silopterus, and that's the one morphologically and in size that I'll be talking about today, most closely related to what I've got in terms of a fossil. So the fossil I'm going to talk about today comes from the earliest Eocene of Wyoming in the Willwood Formation. And it's represented here by a wing and the tip of a bill and a few other bones like toes. I'm going to refer to it euphemistically as Napoleon to allude to its small stature relative to other Forest Racids. But it was a terrestrial predator. And I looked at its phylogeny using 2 different data sets. The first one includes non-Caryamiform outgroups. And you can see that Napoleon there groups within the Caryamiformes. The second doesn't include non-Caryamiform outgroups, but it nests Napoleon within the Forest Racidae rather than just sister to it. So Napoleon was flightless. The primary feathers that are responsible for producing most of the propulsion in flight and flying birds arise from the carpal metacarpus distally on the wing, as well as finger bones, whereas secondary feathers provide lift, and they arise from the ulna. And if we compare the relative lengths of the carpal metacarpus, ulna, and humerus in a ternary plot, we can easily separate flightless and flying birds from one another for the most part. And you can see that Napoleon groups within the flightless birds and outside of the range of the flying birds shown there in black. If we compare it directly to other Caryamiformes, Seriama is capable of flight. Silopterus probably was not. Paraphysornis is one of the true giants that I showed in the very first picture there. Clearly a flightless bird. Napoleon is about halfway in between those two. So what are the biogeographic implications here? If Forest racidae are South American endemics and flightless, then how did Napoleon arrive in North America, since North and South America were separated from one another until the Great American Biac Interchange about 9 million years ago in the Miocene? Okay. So two hypotheses are that they came from South America through Africa to Europe and then subsequently to North America, or from South America directly to North America via the Proto-Antilles. So evidence for this, for the first, comes from fragmentary fossils, none from isolated bones, none of them complete. One from Algeria is a large flightless bird, Lavicatavus, and another a fragment of a pelvis from Europe, Eleutheriornis. Problem with these is that they are considerably later in time, younger fossils than Napoleon. And the next possibility here is coming directly from South America via the Proto-Antilles as part of the late Cretaceous Paleocene Mexican Plateau Senecron. But this model was derived primarily from insects, which would be considerably more vagal than a large flightless bird that had to eat regularly, of course. So there are a number of other alternate hypotheses. They're not necessarily all alternate, and that they're not necessarily mutually exclusive of one another or the former, but they also may provide some contributing explanations. And those are the modern endemisms as we see them today, are relictual refugia in among birds. Other possibility is that there are, in fact, Caryamiformes that are not forest rachids that were present in the early Paleogene or the middle Paleogene of North America and Europe. They're somewhat later in time. There's a possibility that the ancestor of Napoleon flew and became flightless independently, secondarily, or that our time trees underestimate neo-avian divergences, or last, that Napoleon simply misidentified as a forest racid. So the evidence for the first of these is that if we look at the closest relatives of bird fossils from the early Eocene of North America and Europe, their closest relatives are very often present in southern land masses in Africa and Madagascar, transatlantic, nothing like modern day distributions. And the idea behind this was that there was a retraction of paratropical or pantropical forests due to global cooling in the Oligocene. More recently, people have been pointing more towards trophic interactions or other ecological factors to explain this. But either way, the hypothesis fails to address the original dispersal of the birds. So how do we do that? There are two hypotheses here. Both of them assume that Caryamiformes arose in South America, one in which bath ornithids, which are relatively commonly preserved from the middle to late Eocene of North America, came directly from South America up there later on, and that the idiornithids, which were almost certainly capable of flight, came directly from South America to Europe. And then one way or the other, bath ornithids moved to Europe, or idiornithids moved to North America. Okay. Now, it's quite possible that flightlessness was evolved secondarily in Napoleon. Independent loss of flight has occurred in at least 15 different orders of birds. And in some families, it's occurred over and over and over again, for example, the rails. So we have to consider that possibility. One other possibility is that our time tree estimates are faulty. Phylogenomic time trees agree with one another to the extent that the origin of Karyamiformes occurred as part of a great radiation of neo-aves after the Cretaceous Paleogene boundary. But if it in fact occurred earlier on, there would have been less of geophysical barriers to their dispersal. And we have to remember that these time trees are nothing more than estimates. They're hypotheses, and they're sensitive to priors, and as well as the fossil calibrations. And the fossil record of birds is actually relatively poor, and it's known almost exclusively from North America and Europe with regard to early Paleogene birds. So, you know, I do endorse this time tree, but we have to remember that it's always just a hypothesis. So the last possibility is that Napoleon is simply misidentified and it's not a forest rachet at all. And putative synapomorphies, characters that unite Napoleon with forest racety. You know, I only have, for the most part, the wing to work with. And this is a flightless bird, but many of the characters of the wing that are supposed synapomorphies are in fact associated with flightlessness. So I just want to talk about two among many of these, but point out that since they are associated with flightlessness, they are subject to convergence, and that they might misguide phylogenetic inference. So the first of these, Napoleon, is on the black background with two Silopterus forest rachids on the right and the left. And there's a platform-like insertion of what has been identified in literature as that of the Coracle humoralis caudalis. And I'm going to highlight this blinking back and forth here. It's a large raised platform thing, and this is not found in anything else to the best of my knowledge. But this is closely associated with what highlighted in red there, which is the insertion of the pectoral muscle. And in flightless birds, we know from a diversity of them that the insertion of the pectoral muscle moves from a more dorsal position on the humerus down to a more ventral position. And these two structures, you know, move together where they exist at all in birds. And I noticed what we refer to as a ground cuckoo. These birds they're capable of flight, but they're terrestrial and they are predators, and quite rare. But it has a similar structure here associated with the pectoral insertion. And if we could just imagine that this bird eventually became completely flightless and that these structures moved into a more ventral position, it might end up looking very much like what we have in the forest rachids. So one more character that I'll talk about here is the relative proximal extension of the ventral tubercle and the caput, or in other words, the head of the humerus. So the caput is the joint on which the wing moves, flaps, and the ventral tubercle is a point of insertion of rotator muscles. And in birds that are flightless, the ventral tubercle extends further proximal, relatively speaking, to the caput. So we have a restricted movement of the wing. You can see in the Seriama that's clearly not the case. All right, to illustrate this, I'm comparing on the left a New Zealand wren that is capable of flight and recently extinct. This is the Stevens Island wren of lighthouse keeper's cat fame that was flightless. And you can see that the ventral tubercle here is somewhat further proximal relative to that in the flying, the campescidid there. Okay, so in conclusion, I just want to say that Napoleon seems to have been a forest rachet. It probably was. It was definitely flightless, but its biogeography is problematic, excuse me. And it has possible implications for opportunities for dispersal, both for birds and other organisms, convergent evolution and the use of those characters in phylogeny reconstruction, as well as neo-avian divergence times. And for these reasons, this is why Napoleon is significant. So thank you very much. And if anybody has any questions, I'd be happy to take them.
Thanks, Peter. I think just to keep us on schedule, we will move to the next talk. But if someone has a question for Peter, they should put it in the chat, either to everyone, or you can send a direct message to Peter.
Thank you very much.
Okay, our next speaker is John Benning.
Thanks so much for coming to my talk today. I apologize, my camera doesn't seem to be working. But my name is John Binning, and I'm excited to tell you about some recent work I've done along with my collaborators, Dave Moeller and Lex Faulkner, tries to gain a more mechanistic understanding of rapid evolution in response to climate change. And so for the more long-winded version, you can also access the paper that just came out using this QR code. So as environments change through time due to climate change, we can anticipate that there will often be changes in selective environments. So phenotypes that were favored historically, like a plant having large leaves or a bird having a certain color of plumage, may not be as suitable in this new environment, right? So, and for quantitative traits, we often model this as changes in the optimal phenotype or the optimal trait value for an organism through time. And this change could be gradual due to something like increasing temperature that may, for example, increase like higher values of traits like thermal tolerance. Or the change in optimum could be sudden due to something like a drought. And in plants, for instance, we've seen repeatedly that droughts like this select for earlier flowering phenology. And whether the changes are gradual or sudden, populations can respond to these shifts in optima in several ways. So one way is through migration, moving to new areas with more suitable conditions. They could also respond via plasticity. But given the fact that both dispersal and plasticity are often limited, persistence of populations may require adaptive evolution toward that new trait optimum. So let's consider a scenario where the phenotypic optimum is at some historical level, and then a drought begins and the optimal trait value changes. And what we're interested in is how closely the realized mean trait value in the population aligns with that new optimum. And let's assume that historically the population was close to the environmental optimum, meaning that the optimal and realized trait values were very similar. However, after the drought begins, we get a large difference between our optimum and realized trait values. And we can think of the gap between the optimum and realized lines as the degree of maladaptation, with natural selection working to make those two lines converge again. And the greater the degree of maladaptation, the lower the mean fitness and growth rate of the population. And the persistence of that population depends on how quickly the population adapts to that new optimum. And we've documented rapid evolution like this repeatedly in natural populations. So we've observed changes in traits like plumage color in owls, body size in anoles, and phenological shifts in plants. But even though we can document rapid evolution quite well, we still can't predict it. So geographically proximate populations often respond really differently to the same environmental perturbation. Some populations evolve and some don't, even during extreme environmental events. So even though we know that rapid evolution can occur in response to contemporary change, we lack a mechanistic understanding of what underlies the variation in evolutionary responses among populations and species in nature. Now, luckily, we have a pretty good theoretical understanding of the factors that should mediate responses. And these largely fall under the umbrella of the interplay between demography and genetics. So with an extreme environmental event, we may see large reductions in population size, which could lead to genetic drift, potentially overpowering selection and preventing adaptation. Whereas in a larger population, selection is expected to be more effective. And we also need to consider the amount of genetic variation available in the population. So if there's limited genetic variation, you might see a constrained response to selection. Conversely, if there's a lot of variation in important traits, you could observe a rapid response to selection. And lastly, migration and gene flow could have either positive or negative effects on adaptation. So gene flow could increase genetic variation, potentially benefiting that recipient population, but it could also swamp locally adaptive alleles and hinder rapid evolution. And though we typically think about gene flow in terms of spatial migration, for many plants, we can also get gene flow through time with seed banks. So when plants produce seeds and those seeds can survive in the soil for extended periods, this introduces gene flow through time between generations that may have experienced very different selective environments. And let's quickly walk just through how this works. So let's imagine our annual plant population harbors two alleles, P and Q, each producing different phenotypes. And the optimal trait value is near the phenotype produced by allele P, making P more prevalent. And the mean trait value is near this optimal value. So each generation seeds produced by these plants enter a seed bank at the bottom there, from which the next generation is recruited. But then if the environment changes such that now the optimum is near the phenotype produced by allele Q, we can get a lag in adaptation due to continual recruitment from the seed bank, which is mostly full of P individuals that were selected for under the historical selective environment. And there's really cool theoretical work demonstrating this phenomenon, but a real lack of empirical work, despite the ubiquity of seed banks in natural populations. So what I wanted to try and do is to gain a more mechanistic understanding of rapid evolution in wild populations experiencing climate change. And I did this work in the California native annual Clarkia xantiana, subspecies xantiana, which is nearly distributed but locally abundant in the southern foothills of the Sierra Nevada in California. And the black dots on this map show the 30 populations we've been monitoring for about 15 years. And for this study, we focused on 2 of those populations, KYE and S22. And we were specifically interested in the potential for rapid adaptation of these populations during the recent California megadrought. So this was one of the most severe droughts in recorded history. And this 2020 paper was able to attribute about 40% of the soil moisture anomaly during the drought to anthropogenic forcing, meaning that the drought severity was in large part due to anthropogenic climate change. And the most severe period of the drought for our populations was 2012 to 2015, when populations experienced about a 50% decline in precipitation. So while we did want to know simply did evolution occur in these populations during the drought, we also wanted to test specific hypotheses about what might be mediating evolutionary responses. So to do that, we took an integrative approach using a set of complementary methods. To quantify phenotypic evolution, we used a resurrection experiment. And this is an approach you can take with any organism that has a dormant propagule stage like seeds. It's also been done with things like copepods. So we had seeds collected prior to the drought, from the end of the drought, and then 2 years later after precipitation had resumed more normal levels. And then you grow all those up in the greenhouse, and you can directly quantify phenotypic evolution of multiple traits related to drought escape and avoidance. And paired this with long-term data on precipitation and population demography to get an idea of how extreme the drought was in these populations and its effect on population density size. Then used a pedigree design to estimate quantitative genetic variance components so we could estimate additive genetic variance and heritability. And then finally leveraged evolutionary simulations to ask specifically about the role of seed banks in mediating evolutionary dynamics. So looking first at our demographic data, this was a really rough time to be a plant in California. So this plot shows fruiting plant density across time with the drought period in gold. And in both populations, plant density decreased by about 80% during the drought, and survival rates decreased by about 85%. So what this sets up is the potential for a really strong episode of selection. It's accompanied by a large reduction in population size. So then in our resurrection experiment, we quantified phenotypic evolution in a suite of traits related to drought escape and avoidance. But the upshot here is that we only found evidence for the evolution of flowering phenology. So only one of our traits and only in one population, S22. And what we saw at S22 is one of the more common responses of plant populations to drought, if you followed other resurrection studies like this in plants, which is the evolution of more rapid flowering phenology. And this is presumably as a drought escape, works as a drought escape mechanism to finish flowering sooner in the season. So the question becomes, why did these two populations have contrasting evolutionary responses? Was the drought simply not as severe in KYE, the non-evolving population? Well, the two populations actually experienced almost identical precipitation declines, which led to very similar demographic declines. So could population size have limited responses to selection? What we actually saw is that the non-evolving population was estimated to be about 10 times larger than the evolving population, so it doesn't seem like that would be the constraint. And lastly, quantitative genetic analyses indicated that there was plenty of additive genetic variance, and just both in pure magnitude of variance and very high heritability for flowering time in both populations. So this leads us to our final question: what's going on with the seed banks in these populations? So we know from a lot of grueling field work that mostly other people have done in this system that populations of this species differ in their seed bank vital rates. And for the two populations in this study, KYE does have what we would call a stronger seed bank, meaning they have low germination rates and high seed survival rates, so those seeds are staying in the soil a long time. While S22, the evolving population, has a relatively weaker seed bank with higher germination and lower seed survival. Now, the problem is that there's no way to empirically test the notion that differences in seed bank vital rates resulted in contrasting evolutionary responses between these two populations. And the fact that it's so hard to manipulate the seed bank in wild populations is indeed why we have so few empirical tests. But what we do have are individual-based models, as well as methods to test and parameterize those models with empirical observed data. So I built a model simulating a population that experiences an environmental perturbation with the demographic dynamics in the model directly linked to our observed demographic data. And then using approximate Bayesian computation, I asked first for each population whether a model with or without a seed bank fit our observed data better. So what we're looking at in these plots are the distributions of trait change in both populations under two scenarios, either with or without a seed bank, and comparing those distributions to our observed change in flowering phenologies that we actually measured in the resurrection experiment. So greater evolutionary change is toward the left side of the plot here, so you're evolving faster flowering phenologies to the left. And we see that for both populations, the seed bank model was much more probable because our observed data fall much more squarely within the distribution of those seed bank model posteriors. And those seed banks constrained evolutionary responses relative to models without seed banks, which is exactly what we would expect. So then we use those empirical data on phenotypic evolution and population demography from the field to estimate our model parameters of interest. So seed germination rates and seed survival rates. Those are the tiny inset plots there, which together describe the strength of the seed bank. And in these plots, we're looking at the posterior distributions of those parameters. And we see that while both populations were estimated to harbor seed banks, which, as we know, they do in nature, these analyses suggested that KYE, the non-evolving population, had a much stronger seed bank, which is in accord with our field estimated seed bank patterns. And we can see how this relatively strong seed bank produces an adaptation lag, like I introduced in the beginning of the talk. So the stronger seed bank of KYE leads to more maladaptive gene flow from generations that experienced historical selected environments, which leads to a much slower change in the mean phenotype of the population after the drought begins compared to the S22 population, which can more quickly track the change in optimum. So in general, I hope this work and others like it help us move toward a more mechanistic and predictive model of rapid evolution in wild populations. And specifically, I think seed and germ, not just seeds, but germ banks in general, may be an underappreciated constraint on evolutionary responses to environmental change. And there's lots of potential for exciting work here on how these seed banks may introduce trade-offs between demographic and evolutionary rescue during extreme environmental events. So the demographic consequences of seed banks have been studied a lot more than the evolutionary consequences, and how those two kind of trade off during periods of environmental change is kind of the next step I'll be looking at here. So to end, I'd like to thank my co-authors, Dave and Lex here in the field, my mentors and research assistants and funders, and I'd be happy to answer any questions if there's time.
Thank you. Yeah, we do have a little bit of time for questions, and we have a couple. So here is Hezekiah. You want to go ahead?
Hello, can you all hear me?
Yep.
Yes.
Perfect. I was wondering if during this study, any of the microbiological consortia or rhizosphere consortia was sampled to see if they were also changing along with the plant to see if they were meeting those new pressures that are associated with the drought.
I know. I love that question. Unfortunately, no. we have done some work with plant microbe interactions in this system, and I haven't published this, but I do have some evidence that they can. So basically, soil microbial communities can influence plant phenology, which obviously, as you probably know, has been shown in some other systems, and we see a little bit of that here. Yeah, that would have been a really cool thing to do. We collect seeds every year, and so I think in these populations, in all 30 of them, and so I think it would be great to actually start a long-term soil collection as well to kind of go alongside. So then we could ask questions kind of along those lines. It's a great idea.
Thank you.
Let's sneak one more question in from Daniel.
Hi, yes, just quickly, I was thinking of C. Frank's original paper and how the difference in drainage and soils had a big effect. And I wonder if there was some just difference in water drainage between the sites that could also be important in addition to the seed bank.
Yeah, totally. They do have different soils. So the KYE population actually loses water more quickly. So it's sandier, so it's kind of granitic derived soils. The S22 population is on a metasedimentary soil that can actually hold more water, but it is harder for plants' roots to extract water because there's so much clay in there that holds onto the water. Yeah, so it's unclear how that actually played out in terms of plant available water, and that would have been nice to know during the study. But the unfortunate thing is, we're going to see droughts, a lot of droughts going forward in California. So we can think more closely about kind of the environmental, more mechanistic environmental aspects of it going forward.
All right, thank you. If you have any more questions, send a direct message in the chat or reach out to Sean. Our last talk of the session is Jabe Forney, if you're ready to share.
Yes, I am. Okay, hopefully you can see my presentation.
Yep, looks good.
So my name is Job, and I'm a postdoc from the University of Bologna, Italy. And today I'm going to talk about the shared signatures of the shift to endosymbiosis in Enterobacterales. So, it seems I cannot move my slides. Okay, here it is. So we all know that lifestyle profoundly affects the genome evolution of bacteria. And there are few lifestyle which have a stronger impact on genome evolution of bacteria as endosymbiosis. So endosymbiosis in bacteria comes with some relevant signatures, among which we find like a very high AT-rich base composition and accelerated rate of sequence evolution, and most notably very reduced genomes, highly reduced genomes. And well, we also know that bacteria exist in this state of flux of genes. So they continually undergo gene loss, horizontal gene transfer, gene duplication. And we have been interested in understanding how these processes were altered by the shift to endosymbiosis. And well, regarding losses, there's a lot of prior knowledge, which and several lines of evidence suggest and retrieved that across the shift to endosymbiosis, there are two main phases. The first, which is often referred to as turmoil, which a lot of gene loss happens concomitantly, concurrently to the shift in lifestyle. And then there is like a much more gradual genome erosion, like a slower process. And so we were interested in seeing whether this pattern could be retrieved consistently across many multiple and independent shift to endosymbiosis. And we also wanted to assess how gene duplication and transfer were impacted by the change in lifestyle. And for this purpose, we focused on Enterobacterales. And well, we did so because previous literature found many shifts to endosymbiosis. So we started by trying to build a comprehensive and, if possible, reliable systematic phylogenetic framework for the species. And we consider over 200 genomes of Enterobacterales species. But this was a kind of a difficult task because systematic Enterobacterales present a lot of systematic biases, phylogenetically wise. And for example, well, the first problem is missing data. So the fact that there is a very high disparity in gene content, especially between endosymbionts and free-living species, but also the fact that endosymbionts have a very strong anti-bias and accelerated substitution rates is like something which rejects the common assumption with most phylogenetic models assume. So we tried to implement, like to counteract for these biases by employing a wide range of methods. And you can see them here categorized into like major approaches. The first of which is concatenation of single copy genes, which was performed using IQ3, and the second approach, which is influence on gene families, which for which we leveraged species RACs. And then on top of these two approaches, we tried to implement some methods, some methodologies to counterbalance systematic biases. And they were mainly amino acid recordings, so SR4 recording, and which is like a recording of amino acid based on their physical chemical properties, and mixture models. And also like the combination of SR4 recording and mixture models. And you can appreciate how, like, in concatenation, you just use a fraction of the information which is available, because here we considered 103 alignments, while instead by using species RACs, we used like a much higher number of gene trees, so over 10,000 gene trees. And what we noticed was that, which was already something which was known before, is that by using more complex methods, so methods which could like deal better with systematic biases, we were able to split endosymbiont cluster endosymbiont group in many other independent groups. So here there is an example. On the right, you find like a if we can call it like that, like simple concatenation approach, in which is evident the outcome of long branch interaction. And also you can appreciate like a more complex approach, which is depicted on the left of this co-filo, is able to split a big group in multiple groups. So we ended up with two best candidate trees, one of which is depicted here, and they were a tree which was inferred using species and recorded gene families, and the combination of SR4 recording and mixture models concatenated and analyzed via IQ3. So these two approaches inferred 11 independent shift to endosymbiosis, which you can see mapped here with letters on this tree. And well, for that we used ancestral state reconstruction of phytals. And in the outer ring, you see the consistency of endosymbiont groupings clustering across many different inferences which we performed. And I think we achieved like a good consistency, at least with our the methods which we were expecting to be better able to deal with systematic biases. So all analysis which we performed downstream were performed on these like two best topologies, which were nonetheless able to split endosymbiotic traits more than other approaches. And we did so to have like sensitivity analysis on the impact of different, slightly different topologies on the subsequent outcomes. So we performed the GRAX analysis to infer losses, transfers, and duplication. And we kind of traversed all the nodes in which endosymbiosis was inferred and plotted the loss rates. So you can immediately appreciate that there is like a huge spike in the loss rate concurrently to the shift to endosymbiosis. And while there are few exceptions, like the grade H or grade M, and we don't have really time to explain and speculate on why they present different patterns. It's sufficient to say that grade M is made up of endosymbionts of nematodes, so while all the others are endosymbionts of insects. So this, I mean, is a pretty stark difference. Anyway, overall, the pattern is this. So this is highly consistent with the turmoil hypothesis, as expected. But then, like, we excluded all the branches in which endosymbiosis was inferred to have been evolved, and we compared endosymbiont and free-living species loss rate and found no difference. So this suggested us that gene loss is not like the major driver of gene erosion. And in fact, then we focused our attention on duplication and transfer rates, which you see here mapped on our phylogeny. So it's evident that duplication rates do not account for much, while transfer rates actually do. And we found a significant difference. So overall, I think this this pattern suggests that gene erosion is a process which is not attained by an increase in gene loss in endosymbionts, but instead by a reduced gene acquisition. And in fact, I mean, this is quite expected because endosymbionts are expected to have less chances of performing horizontal gene transfer, which indeed happen, but at a lower rate compared to free living species. And well, then we started to consider also like a functional perspective on functional categories, and specifically considering COG functional categories. And here you see gene loss rates subdivided into COG categories and for separately for endosymbionts and free living species. And what is interesting is that while at large there is like consistently a significant difference between free living species and endosymbionts, for two categories there is none. So, and this is like translation and cell cycle control, which, well, they are expected to be some of the more fundamental processes across the tree of life. So in these categories, we didn't retrieve any significant difference between free living and endosymbiont species. While the more stark differences we found were mostly regarding like cell motility, inorganic ion transport, defense mechanism, something which, again, we were expecting. And we then ranked functional categories for each independent loss, which we inferred, and of course also for free living species. And generally speaking, we found that these rankings are all highly correlated. And well, there are some exceptions, and you can see here like inorganic ion transport again, like has a different pattern in endosymbionts and free living species, or also defense mechanisms in which free living species are less likely to lose genes compared to endosymbiont. But generally speaking, yeah, they're all highly correlated. So to sum up our findings, we retrieved consistently a signature of massive gene loss concurrent to the establishment of endosymbiosis, which is consistent with the turmoil hypothesis. Instead, we found no evidence for increase in gene loss rate associated with genome erosion. But instead, our data suggests that it's the lack of new genes acquisition, mostly via reduced opportunity for horizontal gene transfer, that is driving the genome erosion of this bacteria. And as from a functional perspective, most, if not all, different endosymbiont clades were correlated in the ranking of loss of each category loss, and this happens also with free living species. I think it suggests that there is not like a different pattern functionally wise happening in endosymbionts, but instead what is happening is like just a very kind of strong in at least some moments of their evolution acceleration of a pattern which is functionally shared with free living species. So thanks for your attention and for giving me the chance to present this work, which is co-authored with Jacopo and also supervised by Matteo and Dario. By the way, Jacopo is giving a great talk on imidazolite in bivalves in the Bioinformatics Symposium. So if you want to see something more, I highly suggest it. Thank you.
Thank you, Joa. I think we have time for one question. If there's a question from the audience, either in the chat or you can raise your hand. Can't see because my screen looks to be frozen. Is there anything there? Well, unless someone else can see a raised hand, let me ask one real quickly. The loss of gene transfer in the endosymbiont lineages, how much of that is lost because they don't, is there a way to parcel out how much is lost because they don't need it and how much is transfer into the host cell?
Well, actually, it's a very nice question. We didn't consider that. And I don't know if that is really possible with our dataset because we don't have the always the genome of the species from which they've been isolated. So it's difficult to assess whether these genes are kind of lost forever or they've migrated into the host genome. But it's interesting point of view.